So in this video, we're going to talk about Koch and what he did in the field of microbiology and epidemiology. Oh, sorry. All right, so Koch Koch, what he did was he came up with a method for determining what microbe caused what disease. He came up with a series of postulates that are used today still. In doing this, he also was one of the individuals who helped produce mechanisms for isolating bacteria, agar plates, which we use a lot, which in our class we've used throughout the semester to grow specific organisms. And we can use for identifying structurally what organisms look like. So his postulates, there are 4 basic postulates. The first one is the suspected pathogen has to be present in every case of the disease. And that same pathogen cannot be found in healthy individuals. The second postulate, the suspected pathogen has to be able to be isolated and grown in a pure culture. That pure culture then, in the third postulate, the suspected pathogen must infect a healthy test subject, and that test subject must develop the same signs and symptoms as the initial individual or organism in postulate 1. And finally, the pathogen must be re-isolated from the new host and must be identical to the postulate identified in postulate 2. The pathogen identified in postulate 2. It's hard to say pathogen and postulate that many times. So let's look at how he did this. He worked with rodents. And so he would take a healthy organism and infect it with a specific pathogen. That healthy organism would become diseased and would be ill. They would then look for that agent. So in a healthy organism, they would have no causative agent. But in the diseased organism, they would find the causative agent. So here we have a healthy host. Prior to infection, they check, and there's no pathogen. They infect the host with the pathogen. The host becomes very ill, and they find the suspected pathogen in the host. They then grow the pathogen and isolate the infective agent, the potential infective agent. They reinfect a new healthy host. That healthy host also must become ill, and they must be able to find the same pathogen and produce a pure culture from it. If these 4 postulates occur, then you have found the infective agent for a specific disease. Now, these don't work in every case of a disease because there are some diseases in which we can have healthy hosts. There are also some diseases in which we cannot grow in a pure culture. So they don't work 100%, but they work for the majority of our diseases that we find. So now that and this is still the mechanism we use today to identify pathogens that cause specific diseases. So the term pathogen means an infective agent. Pathogenicity then is the ability of a microbial agent to cause a disease. Okay? And the virulence is the degree to which an organism is pathogenic. So higher virulence means they are more pathogenic. And in general, the virulence is going to determine how much of any given microbe you need to establish an infection. So the infectious dose is the number of microbes necessary to cause disease. And this infectious dose can vary within the same organism depending on where it gets, where it is infecting, and between different organisms. And so between different organisms, here's an example. We have Salmonella here, and we have Shigella here. Salmonella has a smaller or has a lower virulence. And because of this, we need more cells to cause the same disease. Shigella needs very few cells, so it has a higher virulence. So salmonellosis and shigellosis both cause severe diarrhea. And so they cause the same disease, but the amount of the infectious dose differs between the two. The median infectious dose, or the ID50 infectious dose, is the number of pathogenic cells or virions required to cause an infection in 50% of a population. So if you have a population of rodents and you put out some microbe, the amount that would cause infection in 50% of those would be the median infectious dose, or the ID50. Median lethal dose is the number of pathogens or virions that is required to kill 50% of an infected population. And so here we see that here we have viruses up here. The norovirus, the infectious dose, so this isn't looking at lethal dose, this is looking at infectious. And this is your table from the textbook. The norovirus needs 1 to 10 virions to cause disease, whereas the rotavirus needs 10 to 100. Hepatitis A needs 10 to 100. So norovirus has a higher virulence than rotavirus or hepatitis A. Here we have E. coli. There are different types of E. coli. And depending on which type, you're going to need a different dose. Here we have Salmonella. Here's Shigella. Here's Vibrio, and I think that's Vibrio parahemolyticus. So you can see their infectious dose and how they change, okay? And then down here we even have protozoans. So then we look at what is a primary pathogen versus an opportunistic pathogen. A primary pathogen is one that causes a disease in a host, regardless of the host's residence microbes or the host immune system. So a primary pathogen causes a disease. If you are a healthy individual and you get sick with some disease, it's a primary pathogen that causes that disease. An opportunistic pathogen, or often also known as a secondary pathogen, causes disease in situations in which the host is already compromised. So this could be part of your own immune system, or not immune system, your own microbiota. Opportunistic pathogens take advantage of the scraped knee, or of the damage, the internal damage caused from, I don't know, jumping off of a 5-foot ledge and breaking your knee, whatever it is, right? These opportunistic pathogens don't typically cause disease, but they will because the barriers are missing or the immune system is already compromised. I'm going to actually stop because the next part is the 4 stages of pathogenesis, and this has been 10 minutes, so I like to keep my videos relatively short in general. So we'll get into the next video talking about the stages of pathogenesis. Bye.